why don't we get started then? It's my pleasure to introduce our next presenting guest, Dr. Catherine Stamen-Breen, CEO, and Dr. Vic Meyer, CSO of Chroma Medicine. The format for this session is going to be a presentation followed by some Q&A, but we'll start by chatting as we get the slides set up. So thank you both for joining us today. Maybe you can start off by telling us a bit about Chroma.
Terrific. Well, we're really pleased to be here today. We're excited to tell you a little bit of background about Chroma and some of the exciting progress that we've made over the last couple of years. So let me start by saying when Chroma was founded, we envisioned a different kind of genomic medicine company. We envisioned one that is leveraging the epigenome. Now, the epigenome is the endogenous system that precisely and durably regulates gene expression. And it does this without cutting or nicking the DNA. It does this without changing the sequence of the DNA. So how does it do this? It lays down a set of methyl marks on the DNA. It does this above the DNA. These methyl marks are responsible for determining which genes are silent and which genes are activated. If you think of the DNA as the hardware, then the epigenome is the software. It's how all of our cells, despite having identical DNA, have very different phenotype and function. This mechanism is durable, and we think this durability will allow us to develop therapeutics that are one-time lifelong cures for patients with serious illnesses. We also think it will open up an enormous indication space. If you think about it, almost all diseases are the result of abnormal gene expression, and so our technology can directly address this. The technology is also quite modular, and that allows scale in our technology. The next slide would be a slide showing our fantastic team. We have a terrific leadership team with deep expertise in genomic medicine and in company building. Our founders are truly the who's who in genomic medicine. The technology was built on some work that was done originally by Angelo Lombardo and Luigi Naldini. They published their technology called Hit and Run in 2016, generating some excitement about leveraging this elegant system for therapeutics. This work was built upon by Jonathan Weissman and Luke Gilbert. They termed their technology CRISPR on and CRISPR off. And it was their work, in addition to work by David Liu and Keith Chung, that formed the basis for the company. We have a fantastic group of investors that have been incredibly supportive. The company was founded by New Path Partners and Atlas Venture. The Series A was led by Cormorant, and the Series B by Google Ventures. This group of investors has been extremely supportive and are excited about the progress that we will hopefully walk you through with some illustrations in a minute. Let me maybe spend a minute telling you a little bit more about how the epigenome works. So the gene expression is really regulated. Oops, I think, well, maybe not. We're still...
You talk myeloid.
Yeah, we're still talking myeloid. Here we go, just in time. Ultimately, gene expression is regulated by chromatin structure. Thank you. We'll swap roles here. There we go. When a gene is tightly wound, it's highly methylated, and this significant methylation results in a closed and inaccessible gene, and that gene is inactive. In contrast, when it's unmethylated, the DNA is open and accessible, and that gene is active. We leverage this system for our therapeutics. The next slide highlights the central thesis of the company. And so when we transiently apply our epigenetic repressors, the target is methylated, and when that target is methylated, it becomes closed and inaccessible. In contrast, when we transiently apply our epigenetic activator, that demethylates the target. The gene is then open and accessible, and the gene is active. This allows us to make a change in the phenotype without a change in the genotype. Next slide. Our technology is also quite modular, as I mentioned. There's 3 components to the technology. There's a DNA binding domain. This is what, of course, precisely localizes our effector domains to the target sequence of choice. And we have some flexibility here. It can be a dead Cas9, it can be a zinc finger, it can be a tail. There's a transcriptional effector domain. This is what transiently represses or activates the target gene. It's active when the construct is only when it's present. The methylation or demethylation effector domain, this is what the company focuses on. This is what our technology focuses on. This is what durably silences or activates the target gene. Those methyl marks that are laid down are durable for the lifespan of a cell and through cell division. Next slide. We think that there's some significant mechanistic advantages to our technology. As I said, this is single-dose gene regulation without cutting or nicking the DNA. And by leveraging this endogenous system and not having a need to cut or nick the DNA, it addresses a number of the well-known risks of gene editing therapies, including risks of translocations that stack themselves on top of each other when you try to do things like complex multiplexing. This is a sweet spot for our technology where we can target multiple genes. We can silence multiple genes, activate multiple genes, or some combination. And we think that there's some very significant use cases here, particularly for more complex diseases. And as we talk about our technology and our future uses for the technology, you'll see more and more multiplexing. And then finally, we can hit almost any gene that's transcribed on the genome, unlocking, for example, non-coding RNAs. And then, of course, finally, the long arc of the company is as we understand the role of changes in the epigenome or abnormalities in the epigenome. Between that and disease, we're going to be very well positioned to address those diseases. So with that, I'm going to turn it over to Vic that will walk through some of the really exciting progress that we've made since the company was founded.
Yeah, thanks, Catherine. It's quite a special day, I guess, to be presenting. Not that it isn't always a special day, but the naturally occurring CpG methylation was actually the inspiration behind the work that led to the Nobel Prize that was awarded today. So it's kind of nice to see how some evolved sort of biology ultimately led to hypotheses that led to some great discoveries that we've all really benefited from in the case of this year's Nobel Prize. I was looking forward to presenting without slides because talking about data without slides is always awesome. Because when you say, as you can see in the following figure, that's a big bar and a really small bar. But instead, I'll have to use our own data, which actually looks quite good. So maybe start to tell you a little bit about what we're going to share with you. So when we set out to develop therapeutics with the technology that came out of Luke and Jonathan and ultimately Angelo and Luigi's labs, there were a couple of sort of key questions, right? The key questions, and we get asked these all the time by investors, so maybe we'll start by sort of posing those questions is, you're not changing the DNA sequence. Is that actually durable? That's sort of question number one. And question number two is that specific? Are you going to be like willy-nilly changing gene expression across the genome if you introduce this DNA methylation technology? And so we wanted to build programs that would allow us to ask and answer those questions in sort of a really prudent, expedient way, while also launching programs that really highlighted the advantages of being able to modulate gene expression without cutting or nicking the DNA. So really, we launched two programs at the outset. PCSK9, I'm sure you all understand that target. It's got a really well-validated preclinical system. Non-human primate locus is very well conserved and allows us to sort of lay down the foundations for how the technology works in vivo with that as the focus target. We launched almost at the same time hepatitis B. HBV is a very difficult target to go after with a genomic medicine because as you cut or nick the DNA, you run the risk of causing translocations across the genome. You run the risk of increasing viral insertions, which has actually been demonstrated by some of the other companies. So we wanted PCSK9 to essentially inform hepatitis B. So let's talk a bit about durability. This is an ongoing experiment. This is, let me just describe this experiment to you. This is a human transgenic mouse. It contains the 65 kb fragment of the human genome, including the PCSK9 locus and its control regions knocked into the mouse. The mouse expresses human PCSK9, and we can monitor the levels of PCSK9 in circulation in this mouse. We applied either our epigenetic editor or controls in a single tail vein injection formulated as an mRNA guide RNA combination in a clinical stage LNP, which we had in-licensed or have in-licensed from Acuitas. And then we just monitor expression of human PCSK9 over time. So a couple of key controls. You can see the vehicle control. You can see the wild-type Cas9 cutter, which targets the open reading frame of the Cas9 protein and gives us about 90% knockdown, which is stable over time. You can see an important control for us, which is CRISPR-I. So CRISPR-I is a catalytically inactive dead Cas9 that contains only the CRAB domain. So this would be expected to repress only when the construct is present. When the construct's no longer present, you would expect it not to suppress. And this is sort of the basic technology that some other companies are using via a constitutive expression mechanism. Then the important epigenetic editor contains not only that CRAB domain, but also the DNA methyltransferase. So it lays down, at least theoretically, it lays down a swath of methyl marks, which are the repressive marks that are then sort of recognized and maintained by the endogenous system. So what you can see pretty clearly with this data, we're now out to the latest time point was day 252. We have essentially complete repression of human PCSK9 after that single administration of the epigenetic editor. So how about methylation? I mentioned methylation. This is a look at what methylation at the locus looks like. So these are mice that were sacrificed at day 28 in a parallel but similar experiment. You can see across the first row, so this is a 5 kb fragment of DNA, where each black bar across the first row is the presence of a CpG dinucleotide. So this is a potential methylation target for our enzyme or a methylation target that would be in the endogenous, would be present in the endogenous genome. You can then see in 2 rows the methylation status in 2 different cell types, liver hepatocytes, which do express PCSK9, and neurons, which don't. And so what you see is a pretty clear difference in terms of the level of methylation in neurons as opposed to hepatocytes. Again, neurons, you have high methylation around that central red boxed region. They do not express PCSK9 where hepatocytes do, and you can see it's sort of a clean landing pad of no methylation. And then each line below that is, excuse me, is an individual animal. You can see 4 vehicle-controlled animals, which give you the baseline. You can see the Cas9 nuclease cut animals look just like vehicle as expected. CRISPR-I, as I mentioned, no DNA methyltransferase, looks pretty clean. And then the epigenetic editor, which gives us very strong high levels of methylation across that central region. So we know from a proximal sort of PD and from a mechanistic standpoint that the mechanism by which we hypothesize this would work actually is the way this is working. So the second question was around specificity, and as you think about epigenetic editing, or at least DNA methylation, compared to cutting or nicking, looking at the DNA sequence itself is not the place we would go. The place we go actually initially is gene expression. Remember, this is a system that's evolved specifically to regulate gene expression, so our first port of call is to ask and answer the question, hey, when we apply our epigenetic editor, which genes change? We use deep read RNA-seq, we look at protein coding genes and non-protein coding genes, we do the experiments in replicates multiple times, and we look to see what changes. This is just 4 different lead candidates from the PCSK9 program. This is a classic volcano plot, which these volcanoes, thankfully for us, don't look like volcanoes, mostly because the only gene that's changed in the case of these 4 editors is PCSK9. And so the x-axis is fold change in log2 space, and the y-axis is an adjusted p-value, so that we have an understanding of statistical significance. And again, you can see the gene that changes, the only gene that changes in this case is PCSK9. We have shown, I think, recently at a liver meeting for hepatitis B, both epigenetic editors that have no off targets, but also epigenetic editors that have a couple of off targets. Because every time we show this slide, people often say, "Well, I don't believe that. There's nothing else that moves." So it's good to have a couple rejected alternatives, and you can see that poster on our website. Okay, so that was PCSK9. This is also an ongoing experiment. This is hepatitis B. Now, Hep B is a little bit harder to study preclinically, as I alluded to earlier. Mice are not infectable by HBV. They don't have the receptor, and so we have to resort to a couple different mouse models to move towards the clinic. This is a mouse model that has the HBV genome stably integrated into the mouse genome, so it expresses all the viral gene products and actually makes viral particles. And so you can monitor both the viral particles based on the DNA, as well as in this case what we're showing here is S antigen, one of the important antigens that we monitor from, as a biomarker for this disease. And what you see here, again, with a single administration, comparing a Cas9 cutter to an epigenetic editor, we get better than 2 logs reduction in terms of S antigen. And again, we have similar data for DNA, and that's durable over time. We do see a little downward drift in that. People often ask, what's up with the downward drift? We don't know. We don't have an experiment, a subsequent experiment that's gone out this long in time, so we don't know if this is repeatable. The lead hypothesis is that there's a slight fitness advantage to hepatocytes that are no longer expressing the integrated HBV genome. Okay, so that's our in vivo work. The place we've been focusing ex vivo is actually in the cell therapy space, and we've been really working on multiplexing. So I think it's our hypothesis and the hypothesis of many that to really make ultimately successful next generation cell therapies, they're going to require a number of genome edits, right? And again, because our technology doesn't cut or nick the DNA, we can multiplex, we can, and I think what's been demonstrated externally in the academic world is upward of 8 simultaneous edits. We haven't in the company yet attempted 8, but we haven't sort of come up with a product that we think requires 8. So we're working on sort of multiplexing in the sort of 3 to 5 gene range. And we do think this is gonna make a better product. We're not gonna have the on-target consequences of a cut or a nick, and it's gonna ultimately streamline manufacturing because you don't need to do the edits sequentially, you can do them simultaneously. So this was an early experiment that we showed at ASGCT this past year. Here we're simultaneously editing 3 targets. We've genericized them as target A, B, and C. This was, again, an early experiment. You can see we get about 80% knockdown on the left-hand side here of the single 80 or better % knockdown of the single targets. When we multiplex them in the dark blue, we get similar efficiency as with the single targets, suggesting that the system is capable of multiplexing. If we then take those multiplexed edited cells and subject them to a genomic analysis, in this case we were looking directly at translocations between the targeted sites and comparing the epi editor to a triple cutter, so a straight up Cas9 triple cutter. This is a single cell assay, it's FISH. We're looking at 300 cells here, and what we see is frankly what's expected, but it's always rewarding to get what you hope for. The non-targeting control and triple epi silencer essentially had no events or one event that was probably the result of the electroporation. But fully 10% of the cells showed reciprocal translocations across those sites. If you look deeper, and the imaging technology we used allowed us to actually look quite deep at the genome, we saw fully a third of the cells with the cutter had some genomic abnormality that was associated with the presence of the Cas9 nuclease. So you do get significant changes in the genome when you apply a triple cutter that you don't see when you apply a triple epi editor. Okay, so we have taken that work and moved it forward. One of the first cassettes we're working on is an allogeneic cassette, so we're eliminating genes that will essentially prevent expression of MHC class 1 and class 2 on the surface, also eliminate expression of the T cell receptor. And here we're just showing some data where we get, with simultaneous application of the epi editor in comparison to Cas9 or CRISPR-I in the yellow or in the dotted yellow data, we're showing simultaneous silencing of between 90 and 99% of these 3 targets. Particularly satisfying is the T cell receptor, which eliminates graft versus host and is really the safety edit here, not the efficacy edit here. It's really almost knocked out completely, so it's 99.5% knocked out, I think, in this experiment. So we're pretty excited about the cell therapy stuff. We think it's, and I think there's going to probably be a question on partnering. We do think this is broadly applicable and an interesting platform for partnering for folks that are working on cell therapy, both in the oncology space and the non-oncology space. So I think with that, we will stop and take questions.
All right, great. Thank you so much for that. Maybe before I jump into my questions, I'll open it to the audience. Any questions from the audience here? Otherwise, while you guys think about it, you talked a little bit about this already, but in terms of the type of indications that you can go after in the multiplexing that you showed, I think in general in the genome editing space, we're seeing some clustering and of approaches that focus on the targets such as PCSK9 that have been addressed by technologies such as RNAi. Can you expand a little bit on how your capabilities, whether it's including multiplexing or activation, how you're going to be able to go beyond the targets currently addressed by the RNAi space?
So maybe I'll start, and then Vic, you can jump in. So we think the sweet spot for the technology is multiplexing. It's an opportunity for us to address complex diseases and be able to address those diseases with potentially with genes that approach disease mitigation in different ways. Whether that's silencing multiple targets that are in the same pathway or separate pathways, or combining silencing and activation. We think this is something that merely is the sweet spot for this technology and an approach that traditional gene editing is not well suited for.
Yeah, and I just to add on to that, I think the advantage, so we love RNAi, it's great technology.
Yep.
You know, we do aspire, and we think based on the data we have and essentially the biology that's out there, that a single administration should be durable. So we think, have the advantage of durability over RNAi.
And in terms of the activation work, where are you on that?
Yeah, so so just for description, activation is essentially the opposite of silencing, right? For silencing, we add methyl marks, we turn off gene expression. For activation, we remove methyl marks and we activate gene expression. Activation is going to be a little bit more nuanced than silencing because you essentially need two things to happen, right? So with the methyl marks, you actually recruit active repressive factors. When you remove the methyl marks, you remove that active repression. What you need to have happen is to have the right cellular context such that the transcription factors and the other machinery, the signaling machinery is in place to drive expression. So there's a little bit more nuance when you're picking targets for activation. So we have launched one program that requires activation for moving forward, and that's in the sort of lead discovery space right now. Personally, I do believe that technology development is best done in the context of programs, and so we have, as opposed to sort of just going out there and doing activation, we're actually doing activation in the context of an ex vivo program, and we'll be launching, we intend to launch a program next year that requires activation for an in vivo program as well.
Great. And you alluded to partnering earlier on the cell therapy front. More generally, how are you thinking about partnering in cell therapy or elsewhere?
I think we think about partnering in the context of augmenting our capabilities internally, whether that's a technology capability or a capability in a therapeutic area where we don't currently have internal expertise, or, for example, it might be a delivery technology that we don't currently have access to. And so that, we think, is our best options for business development. We're excited about, we remain pretty focused at Chroma. We believe that it's probably better to be, particularly in the early days of a company, to be deep in epigenetic editing and not end up sort of an inch deep and a mile wide. And so philosophically, that's how we've approached business development.
Perfect. In the last 50 seconds or so, maybe I'll ask, what might folks be missing about Chroma and all the discussions that we have so far?
As we were tossing this around this morning, and actually Vic had a nice thought as we were tossing this question around this morning.
Yeah, I guess I would just underscore something that Catherine said earlier. I think what people miss is sort of the breadth of the applicability here, right? You know, when you think about genomic medicines and genome editing, people immediately, I think, and rightfully so, go to genetics that cause disease and can we fix those mutations, et cetera. But really, when you think about drug development and drugs in general, you know, if you can modulate gene expression, I think you can address almost every disease, right? All of our small molecules, siRNAs, et cetera, essentially they're at some level in general modulating gene expression. Maybe it's at the protein level, maybe it's at the mRNA level. If you're able to sort of hit it at the source, at the DNA, I think the applicability here is incredibly broad. And that's one of the reasons we've started out with sort of large indications that will have sort of a large patient population, is that sort of breadth of application and our belief that epigenetic editing is ultimately going to be a safe and really powerful approach.
Great. All right, so I think we're at the end of our session, so I'd like to thank you both again for the presentation and the questions.
Thanks for having us.